Hello, thank you for joining the webinar. We are awaiting few joinees, so you just have to bear with us for a few minutes more and we'll initiate the webinar. Thank you for joining. Hello, I'm Lakshmi Achyuta. I'm a strategic advisor in biotech, pharmaceuticals, and medical devices. I have about 27 years of experience in quality, compliance, strategy, and skill enhancement, having worked previously in biotech, pharmaceuticals, CRO, clinical research organization, and contract research and manufacturing organizations. Welcome to the webinar on effective good laboratory practices in the context of COVID-19. Few housekeeping rules before we initiate the webinar. We request you to stay on mute. We request you to wait for the Q&A session to ask your question. Feel free to use the chat window to drop your questions during the session. In today's webinar, we explore the relevance and importance of GLP in the context of the drug development life cycle. We explore the relevant requirements related to quality of data in GLP and what the impact is on the study conduct. We explore the COVID practices and procedures that are required during the conduct of the study, especially when the study is being done in this period of time. If you were to see the drug life cycle in its entire entity, the drug moves from the drug discovery process to development process, wherein the analytical and process development activity happen, and subsequently it moves into preclinical development, clinical trials, and it moves into manufacturing and further into commercialization. During the entire sequence of activity, the GLP is regulating the preclinical developmental activities, and it has a major impact on the entire drug life cycle. GLP, the acronym, is the quality system for the conduct of non-clinical health and environmental safety laboratory studies to intend to ensure that the quality and the validity of the test data is as per the requirement of the study and the regulatory requirements, to ensure that the studies are planned, performed, monitored, recorded, and reported. So it's very, very critical that the quality and the validity of the test data should be traceable, it should be reliable, and it should be, you need to ensure that it is available during the entire data sets. Historically, the public agencies and the regulatory agencies like the US FDA are responsible to protect their citizens, and the legislations are in place to ensure that the responsibility on the rests on the manufacturers to ensure that the safety and efficacy of the drugs is assured by the manufacturers. So in the 70s, the US FDA identified a lot of inconsistencies in the data generated by the various agencies conducting the GLP studies. A classic case is that of the Industrial Biotest Lab, which was conducting the tests on behalf of Procter & Gamble, wherein over there, what had happened was that the mice had developed cancer and it had died during the conduct of the study. However, the team threw away the dead mice and covered the results, deeming that the products were good for consumption, for human consumption. So this led to major investigation by the regulatory agencies, and finally criminal investigation was launched against the stakeholders, and their criminal penalty was awarded to them. Subsequently, in the year 1978, the US FDA formalized the GLP, which had a worldwide impact. Worldwide impact in a manner that any company conducting the business with US had to follow the US GLP regulatory requirements. This led to various countries having their own regulatory requirements related to GLP. Subsequently, in 1981, the Organization for Economic Cooperation and Development formalized the GLP as an international standard. And that's the reason why, as of today, OECD GLP is a formalized international standard being used by the various regulatory agencies across the world. In 2002, the GLP compliance in India was initiated to be monitored by the National GLP Compliance Monitoring Authority, that is the NGCMA. And in 2011, India became a full member of the Mutual Acceptance of Data, wherein the study conduct would be done in India and the data would be generated in India. However, the reliability of the test data and the test information would be submitted across the globe in various regulatory agencies. The regulations to follow for GLP, the 21 CFR Part 58 as per the US FDCA Act, the Good Laboratory Practice for Non-Clinical Laboratory Studies, and OECD GLP for non-clinical lab studies across the globe. So any company interested in conduct of study data being submitted to US FDA need to follow the OECD GLP, and there are a lot of similarities between the Part 58 requirements as well as the OECD GLP requirements. What are the advantages of having a GLP? It definitely increases the confidence in the data quality and the reliability of the data. Very importantly, there is mutual acceptance of data. That is, the data generated across the globe and submitted to various regulatory authorities is accepted. And it assures that the data that is generated is a true reflection of the results obtained during the conduct of the study. And importantly, considering that the drug development life cycle takes about anywhere between 10 to 15 years, it shortens the time to market for the new products. Objectives of GLP is to ensure that the data submitted are true reflection of the results obtained during the study. Very importantly, traceability of data. It promotes international acceptance of tests. And the elements that are required to address the GLP components within the organization is as indicated. We would go through this one by one, and we would look at the challenges that we would have. The facility and organization, the other element related to personnel and training, documentation, sample identification and management, this is also known as chain of custody, chemicals and reagents, instrument qualification and calibrations. We would look at analytical methods and quality assurance. The facility should be very well planned, very well designed for the conduct of the study. It again depends on the kind of activity being done, whether it's a microbiology lab or it's a biologics lab, or for that matter, if it's a virology testing lab. So the planning of the facility is a very, very crucial component. It has to take into consideration the pre-analytics activity, the analytical activity, and the post-analytical activity, and to ensure that the entire activity happens in the phased manner, in the flow that the activity is happening within the laboratory. So the facility has to be designed to ensure, and it has to be access controlled. Organization of the entire GLP system is a very critical requirement. And the four key functions are the test facility management, the study director, the quality assurance unit, and the requirements related to archives. And it is very important that the study director has to be designated for the conduct of the study. And they are whole and sole responsible to ensure and assure that the study conduct has been done as per the GLP requirements. Personnel involved in the study conduct have to be qualified as per the education, as per their experience, knowledge, skill, and if required, additional training should be provided. For example, if there are any regulatory updates, any regulatory changes that have taken place, then they need to be updated and trained as per the requirement. Analysts have to be certified. Analyst certification is a very important component of a personal training in the laboratory area, based on which analyst certification metrics would be derived in the organization, based on which the analyst would be certified. And this certification would include being given unknown samples, or they are also known as the blind samples. And then only the analyst will be certified. And subsequent to that, then only they are allowed to participate in the conduct of the study and conduct analysis. It is important to note that analyst certification is not a one-time activity. It is a routine activity, and it could be based on the samples being handled by the analyst. It could be done on a periodic basis. The frequency could be as much as once in 6 months or once in a year. So based according to the conduct of the study, or the analyst requirement, they would be certified. The job descriptions have to be very well defined as per the tasks and the roles and the responsibility undertaken by the various team members. Documentation and records. So documents are something you refer to execute an activity, and records are evidence of having conducted an activity. So there is a statement that goes, not documented, not done. So in God we trust, the rest require documentation. So it's very, very critical for you to understand that documentation, be it a paper document or it could be even an electronic document. And in some companies, they use a hybrid approach wherein they use both paper as well as electronic documents. And it's very, very critical to understand that the controls would remain the same for both. The procedures and the protocols that are derived have to be written in a simple language, in a chronological manner, and all in such a manner that a person of education and qualification, equal education and qualification, should be able to conduct the activities without requiring additional support. So it's very, very critical to understand that this has to be followed. It has to be written in a simple language, and the effective version of the procedure has to be in use, and the previous procedures have to be safeguarded and put into archives. And all stakeholders have to be involved in, whoever is involved have to be trained in the conduct of the study. Good documentation practices are in this particular ALCOA principles. The guidelines have been provided by various regulatory agencies, including the MHRA and other regulatory agencies. They are ALCOA Plus. Earlier, it used to be called as ALCOA. It's now called as ALCOA Plus principles. Attributable, legible, contemporaneous, original, accurate. The plus comes in as in complete, consistent, enduring, and available. What do you mean by attributable? Attributable is very, very critical. You need to attribute the particular activity to the data to a particular activity. So the 5W1H principle stays here. The 5W1H principle indicates that the where, when, what, why, who, and how have to be addressed in this particular attributes of the data. The data has to be legible, be it in electronic, be it in paper. The data has to be legible. That means you should ensure that any corrections made are struck off with a single stroke, signed and dated with the corrected data. And very importantly, whitener should not be used. Subsequently, in the electronic data, all changes that has been made has to be traceable. There should be an audit trail available. The date and timestamp has to be available so that the entire activity can be reconstructed. You need to ensure that the data is contemporaneous. You enter the data as and when you do the activity. That is, as soon as you finish that particular act, you need to record the data. The data should be original. The first point of entry of data is known as the source data, or it's also called as the raw data. And this could be in thermal paper. And as you are aware, the thermal paper over a period of time becomes illegible. That means you are unable to decipher the content in the paper. So you need to photocopy them, you need to review and verify it and put your sign on it. And accordingly, the data, first point of entry of data, if in case it's in the lab notebook, you need to preserve the lab notebook. Same thing holds good for the electronic data as well. Accuracy of data. The data that is obtained is what needs to be indicated. That means if in case an experiment has failed, the failed experiment data also should be documented. Not documenting the failed experimental data and indicating it as a past experimental data is actually an instance of fraud. Okay. The data has to be complete in all its respects. That means you need to ensure that the data about data, that is the metadata, is available. You need to ensure that it is consistent to the activity conducted. You need to ensure that it is enduring. You need to ensure that the medium and the data is enduring because it needs to withstand and it has to be stored for a long period and it has to be available for the regulatory agency scrutiny as well. The chemicals and reagents used, you need to ensure that the chemicals have appropriate labels, appropriate safety data sheets are available, and they are appropriately stored in safety cabinets, acid-resistant cabinets if required, and the solutions have to be labeled with the expiry date and the open date. You need to ensure that the deteriorated or outdated reagents are not being used, because in case that you're using it, then it's going to lead to faulty experimental data being generated. So you need to be careful on the type of chemicals and reagents being used. The instruments that are going to be used for the study conduct have to be validated and calibrated. You need to ensure that the personnel who are using it are adequately trained in handling it. And you need to ensure that it is in compliance. That means it has to be in the state of calibration and validation. And all the records and the logbooks or the relevant information related to the instrument have to be maintained. Any deviations, any out of calibration have to be recorded and appropriately handled in the quality system requirement. The data produced by faulty instruments may give an appearance of valid data. So you need to be aware and you need to ensure that they are not being used for the study conduct. Sample management, a very critical component of the study, is also known as and is part of chain of custody. Chain of custody initiates when the sample is generated and it moves through the various stakeholders when it gets shipped, or when it moves from the study conduct area, say the toxicology lab, and then it moves into the area of the lab. The samples have to be verified. The accessioning of sample is a very critical activity, wherein the integrity of the sample needs to be verified. You need to verify whether there's been tampering of the seal. You need to verify if the sample is leaking. You need to verify if the sample is contaminated prior to issuing a unique identification number. Once that is issued and the sample is accessed and it gets into your database, it moves into the sample storage area. The sample storage area is a restricted area wherein the samples are stored, and the sample custodian is the person who's authorized to handle the entire sample storage area. The samples such in the storage area have to be accountable. That means any sample being drawn from the sample vial have to be accounted for. It has to be traced to the activity conducted as to which experiment handled which sample. So there should be a traceability of between the data generated and the sample that has been received. Very importantly, the original source of the data should be recorded and connected with the analytical data. So especially in chain of custody, the entire activity has to be traceable. The entire activity has to be reconstructed in the sample management and chain of custody. When analytical methods are being used, they have to be validated, and the tests that are going to be used have to be developed to ensure that the drug substance or the product and the characteristics are established acceptance criteria for that particular characteristic, and the choice of analytical instrument should be appropriate to the method and the intended purpose and the scope. Quality Assurance Unit is an independent unit. It reports directly to the test facility management. They are responsible to ensure that the quality management systems are in place. The review of documents and records are appropriately handled. They're responsible also for the training. They're responsible for also for the internal audits or in-life inspections or retrospective inspections. They're very importantly responsible to review the investigations that are taking place in case of any deviations or in case of any untoward incidents that have happened or a laboratory incident has happened. So quality assurance is a very, very critical component of the entire GLP scenario. Once the study conduct is completed and the data has been reviewed by the teams and the entire data has been reviewed by the quality assurance and the quality assurance comments have been addressed by the teams, the data moves into archival. Archival is the destination wherein the documents or albeit electronic or paper records are held for a certain number of years. It's again based on the regulatory requirements. And coming to the archival area, it is a restricted area. The archivist is the custodian of the area and it is access controlled, it is physically secure, and the systems or the infrastructure in such a manner, constructed in such a manner that it ensures that it is protected against deterioration. For example, the archival facility has temperature control, is temperature and humidity control, and typically the paper documents are stored in compactors and they are housed in this physically secure area, archival area. And subsequently, the electronic data that is generated, they either move into the server, many companies or many organizations move into the server, or they have backup data. The data that is backed up is typically in copies. So one typically rests within the facility and one rests outside the facility. This is to ensure that the data would be available during the subsequent review of the documents by the regulatory agencies. Why is archival such an important activity? What happens when the regulatory agencies receive the dossier? Once the regulatory agencies receive the dossier, the review initiates and they are likely to come to the facility for inspection after a couple of years. And the archival need has to be ensured in such a manner that the documents are retrieved in a timely manner when the regulatory authorities come in and they verify it. So what actually happens during regulatory agency inspection is that reconstruction of the activities are performed. So the data is already available with the regulatory agencies. And when they come in to the facility, they're verifying whether the data is available at the GLP facility. They're going to verify the study reports. They're going to verify the study files. Subsequently, they're going to move and verify whether the data is traceable, the lab data is traceable to the equipment and instrument being used. And subsequently, from the laboratory area, it goes to the backup data. So they do the entire reconstruction, or you could say they do the entire traceability of data is done. They're basically verifying whether the data is reliable, whether the data is authentic, and whether the data can be traced. Only when these activities or when the data is, they're able to trace the data, will they assure that they come under the assurance that the entire study has been done as per the GLP requirements. So what happens when we have a situation like COVID or the pandemic that we are experiencing? Because some of the studies were done in the cusp of the pandemic. So what happens to the data? How are we going to handle the entire activity? What are the procedures and practices? What are the requirements that we need to address? So that's what we are going to discuss in subsequent couple of slides. The declaration of the pandemic ensued with the government agencies declaring it and providing instructions to the various industries, to the public in general, to the industry. And based on those, the organization had to review that requirement because it's a new requirement that has been come in. It was not there previously. So this is a new requirement issued by the government agencies, which has to be addressed. So the first thing an organization would do is review the current practices, review the current infrastructure with that of the government agencies' instructions or the guidelines being issued by the agency or the regulators. And that's where they would have identified the gaps. The risk assessment has to be done. And the gaps and the risk assessment ensure and assure that the requirements and the practices have to be in place prior to the activity being undertaken. So we review the activities relevant to the movement, relevant to the communication, relevant to the premises, and very importantly, relevant to the lab operations. So it's very, very important that the team within the organization, typically these are known as crisis management teams within the organization, who respond, who have their roles and responsibilities already well defined, who raise up to the occasion and they take on the requirements and they start reviewing the current infrastructure and practices with that of the government agency instructions. Typically, many organizations have restricted employees at any given time. Like for example, many of the employees have been put on shift system, or many of them come in alternate week, or many of them come in on alternate day, like they have this odd and even day wherein the employees have been designated to come in and work. So there's also zoning of activities that are being taking place. And additionally, the first activity that the company typically undertakes is to identify high-risk employees. That is, for example, the pregnant women, older employees. These are the high-risk employees and they have to be taken care of, and typically they work from home. The visitors are restricted, and only visitors, especially related to vendors who have for very critical material, are allowed inside the premises. And they need to undergo the scrutiny wherein they have to produce the relevant tests that they have undergone within the 48 hours that they're COVID negative. They need to get themselves checked for temperature. They need to adhere to the strict requirements of the organization. The organization would have their own particular restrictions and they have to follow all those norms. It's a very, very critical and important activity which the visitors have to take place. In fact, the vehicles coming in also need to ensure that they are following these restrictions. Coming to material movement, none of the materials would directly enter into the lab area or into the area where the study activities would be undergoing. And typically they would be housed in a quarantine area or in a dedicated area, wherein the surface would be sanitized, the materials surface would be sanitized. In fact, when the trucks come in fact, they are also sanitized as well in many instances. The samples have to be surface sanitized before they are allowed to come inside the laboratory area. Very importantly, the waste segregation, the waste generation or the waste segregation done by the teams have to follow the regulations as per the waste disposal norms. It's very critical that employees have to be trained additionally on the waste segregation rules to iterate and to let them know that the segregations that are required in the current situation. And they have to be trained as well in decontamination of the material before it is disposed of. Any spills that has happened, they need to ensure that the spill kit, the absorbent material that is there in the spill kit has to be used. And the minute it is used, it is considered contaminated. So they cannot discard that material directly into the waste. They have to, in fact, decontaminate it, or rather, you could say in the language of many labs, they have to be quenched before it is disposed of into the disposal as per the requirements of the waste norms of the company and the organization. Communication plays a very key role, and as per the organization norms, only the authorized people are responsible for the communication within and outside the organization. And it's restricted to only few people, the top management. The corporate communication team is the one that typically handles the internal and external communication. And employees have to be aware of that. Employees also have to be trained, and they have to be indicated that they cannot be circulating incriminating information or videos, especially on the social media, considering that they are very active on that. They have to restrict the employees circulating of the videos. Any concerns or any issues have to be escalated to the supervisors. There is a mechanism that the communication channels within the organization happen, and that's how the escalation metric should go. It could be, in fact, instance wherein there could be a primary contact as well or a secondary contact. All these have to be escalated to their supervisor and the subsequent head of the department. Considering the pandemic and considering the situation and the impact that it has on all of us, dedicated hotlines are available in many organizations wherein their employees check in and they voice out their stress level, or they could voice out any help that they would require. And this is typically provided by the organization, because at this instance of pandemic, it's very crucial that the employee well-being and their mental health is taken care of. The entry into the premises, again, is only on as is required basis only. Not everybody can come and visit in such a situation. Only people with prior approval are allowed to enter into the facility. They're not otherwise allowed to, they're restricted to enter into the company. And including the employees, everybody would undergo a temperature or oximeter check. In fact, many organizations have provided their employees with a thermometer, and the employees have to check their temperature once in the morning and once in the evening and enter it into their database, so that the organization can get an idea as to who could be a potential carrier. They need to sanitize their hands. They need to wear the masks, social distancing, and precautions in all areas. And personnel are restricted to designated facility and buildings. This is known as zoning. The terminology used in the industry is known as zoning. And personnel are restricted only to that designated areas. And if in case they need to move around, then they are given special badges. And only these designated employees are allowed to move into the various areas. And visitors have to be always accompanied. Vendors have to be accompanied, and they have to restrict themselves only to those designated facility or buildings. Coming to the housekeeping norms, routine practices as per the procedures are followed. Additional practices in general area, especially where there is high footfall, or we could say high risk area where there's not many number of people, like the washroom or the change rooms, the canteen, the lifts, the entry gates. So these are the areas that have to be under consideration, wherein they have to be sprayed daily 2 times. Many of the industry follow this norm, wherein they spray 2 times with isopropyl alcohol or with a suitable disinfectant. And in coming into sanitation, they have increased the frequency of cleaning, especially in areas of high risk, as I said earlier, in the cafeteria area, where there's a lot of people who come in and they could be exchanging in the shift exchange in those kind of areas. That's where they need to ensure that it is frequently cleaned, and materials used either should be sterilized or surface disinfected while it's being transferred from outside areas. Lab operations, as per the companies or the organization norms, typically the access to the area is restricted only to the employees who have to function. And again, employee are restricted. They come in shift system. and typically it runs in 2 or 3 shifts, and they need to remove the street footwear. They need to wear appropriate footwear in the relevant area before they gown in and wear the PPE. And they have to handle the garments carefully to avoid contamination. It's very important that they have to avoid contamination. They need to restrict the movement. They can't keep moving around in the same area because the aerosols are produced and likely to carry the contamination level. Avoid sharing of equipment or tools. Sanitize before and after use. And frequently clean the areas that are used or touched areas, like the door handles especially, that's where it is frequently touched. Do not fill or execute documents together, because many of the activities have a done by, check by, activity. That has to be verified. So you have to be 1 meter apart. You need to ensure that you do not fill the documents together. Avoid touching surfaces, other surfaces like the railings or the window, and ensure that the first aid is available. Coming to further into the nitty-gritty of the lab operations, trained employees need to undergo additional essential containment requirements and practices. The sample received have to be identified and traceable. If in case the sample does not have any identity, or you're unable to trace it, or you feel it is tampered, or if it is a damaged container, do not touch it. Appropriately handle it in a proper manner as per the norms. The biological specimens have to be handled as per the requirement of biological safety cabinets. When it comes, that's the pre-analytics. Once the sample enters into the laboratory area, you need to handle the technical procedures to ensure in such a manner that it minimizes the generation of aerosols or droplets. Appropriate measures to be considered, especially when you are handling aerosol exposures, ice splash during sample processing, infectious material spill, and very importantly, document all deviations in the context of COVID. That is, like in last week, I audited a facility and I observed that the calibration was due in May, but however, the calibration was subsequently done in August. And they had documented it as a deviation, they had appropriately handled it, and they had done a risk assessment. So that's how the activity has to be handled. In case there are any study-related procedures that have been deviated, like for example, the QA didn't conduct the in-life inspection because non-availability of the QA person, or if there is a change in the study director because the study director was unable to come from the area where he lives or she lives. So these are the things that have to be documented, because if you recollect, when the regulators come in, they are going to look at reconstruction of activities. So the COVID instance, the COVID situation would be gone in the next couple of months or probably a year. And subsequent to that, the regulators would be down here reviewing the data. So you need to ensure and assure to them that during this phase of COVID, the conduct of the study was as per the GLP requirements, which we discussed earlier in the session. All those requirements were adequately addressed. In case of any deviations, all these were properly documented and properly addressed. So the traceability in terms of whether it's a paper document or in case it's an electronic document should be available. So it's very, very critical that documentation is very crucial, and more so in the situation of COVID-19. Once the analysis is done, once the activity or the study conduct is done, the proper processes of identification and segregation of contaminated materials should be adopted before disposal. So it's very, very critical that these activities are handled in an appropriate manner. The key takeaways for this is that GLP is an FDA regulation, which is accepted, approved as an international standard by the OECD. In India, it is monitored by the National GLP Compliance Monitoring Authority. To avoid fraudulent activities in testing labs, to save human and environmental health, to facilitate international trade and establish good relationship among the countries. So it is the question and answer time, and we have received a few questions here. So we have a query. Will notes be given? Yes, definitely. We will publish the recording and the presentation. Follow us on the LinkedIn for updates. And there was a question from one of the participant in LinkedIn, wherein they had queried training on GLP with certification. Yes, training on GLP with certification is available. I advise you to visit www.therqa.com, the researchqualityassurance.com. Earlier, it used to be called as ARQA, the British Research Quality Assurance. Now it's called as www.therqa.com. You could also visit SQA website, the Society for Quality Assurance website, that is www.sqa.com. Additionally, you could also explore this in your National GLP Compliance Authority's website, which would be available in your relevant countries or in the place where you are living currently. There was a question related to, will there be a feedback? Yes, you will get a feedback form after the webinar, wherein you could provide your feedback on the session that was conducted. Any other questions? So there is a question from Dr. Madhava Rani. Is that needed to test all drugs in pregnant women all the time? So GLP is a study conduct on preclinical requirements. It is not a clinical requirement. That means GLP studies are done on animal or animal models. It's not done in humans. Clinical studies are done in humans. And when they are conducting clinical trials, definitely pregnant women are not considered at all. In fact, if you look at the clinical phase of phase 1, 2, 3, especially in the phase 1 clinical studies in phase 1, phase 2, the participants are typically male. The women are not considered for the conduct of clinical trials. Is there any specific guidelines for recording Hvac parameters? Yes, definitely they are available. You could verify in the ISP guidelines and you would find them. Any other questions? If we are conducting studies as per the OECD GLP, should we still follow 21 CFR Part 58? Depends on the submission of the data. If you are submitting the data to the US FDA, then definitely you will have to follow the norms of the 21 CFR Part 58 as well. So for example, if you are in India and you are conducting the GLP studies, you need to ensure that the OECD GLP requirements are considered as well as that of the Part 58. There are certain nuances, differences between the Part 11 and Part 58 and OECD GLP that is available. I could share it with you later. You definitely need to follow in case you are submitting data to the US FDA. Regarding the requirements on BSL for lab requirements, currently I don't have the information. Definitely we will post it to you. When the whole issue is on protection of samples, how could coronavirus escape from the Wuhan lab? Who will be held responsible is the query posted by Mr. Raghu MR. Well, this is my opinion. I wouldn't like to get into the discussion of it, whys and hows, just to indicate that controls are in place in any laboratory that is handling live viruses. They would definitely have to follow the international norms. They will have to also follow the national and regional norms. there are instances wherein rogue employees do fraud and misconduct. And I would say this is a directed instance of that particular activity. I wouldn't like to get into that because it has a lot of implications, okay? And it would deviate from the topic. What impact has the pandemic have on the study that was initiated at the cusp of the pandemic? Is a very interesting question sought by Mr. Gopal. Yeah, very, very critical because during, as indicated during the discussion, during the session, the regulators are going to review the data generated after a couple of years. And definitely this data that was generated during the cusp is going to be reviewed by the regulators after a few years. You need to ensure that the documentation is appropriate and there is reconstruction of activity. For example, let me take the case of the calibration, which is the easiest example I can quote. If you take the calibration instance, the calibration was due in May, but unfortunately, because the COVID pandemic and the restrictions declared by the government, the vendor was unable to visit the facility to do the calibration because it was an external calibration and the instrument was an annual maintenance contract. So they came in August, they did the entire calibration and the entire activity was documented. There was a deviation that was taken. So when the regulators come in and when they look at this data, when they look at this instance saying that, okay, the calibration was due, it didn't happen, there was a deviation taken, there was a risk assessment that was done, there was a corrective and preventive action that was taken, this gives an entire picture to the regulators. This gives an assurance to the regulators that in spite of the study being conducted at the cusp of the pandemic, the team was able to handle it. Let's take the example of a study director not available. Did the new study director come on board where the animals being fed on time? So these are the questions that have to be answered. These are the identification points that have to be addressed and documented appropriately so that there is reconstruction of activity. Next question is the GLP not applicable for clinical labs? Again, an interesting question. GLP is for preclinical studies and clinical studies, and the clinical studies come under the GCLP requirements or the ISO 15189 requirements. So GLP is not applicable for clinical labs, definitely. Or it's only applicable for labs related to labs related to preclinical samples generated during animal studies. However, GLP is applicable for bioanalytical labs. That's a different discussion, and I think there is 10 minutes left. And Mr. Venkateshan, I request you to reach out to me so that, or I would reply to you in a separate mailer because this is a separate discussion that we would like to have with you. Next question is coming from Dr. Yeah, I think this I completed. We have 10 minutes left, and I will answer to the best of the time that I have, or else we get back to you on the social media, or we get back to you in person. Is implementation of GLP in lab mandatory or voluntary? This is queried by Mr. Manon. It's important that the GLP studies being conducted for submissions have to be done in GLP certified laboratories only. So GLP in that way is mandatory. GLP in the context of medical devices would definitely take time. I think that this would require a separate session altogether, I would say. Trying to see where I think I got lost somewhere here. Do we need any regulatory compliance for software developed for such labs? Regulatory compliance for software or computerized system, you need to follow the 21 CFR Part 11, that is the electronics record, electronic signatures, as per the US FDA requirements, and also as per the Annexure 11 requirements of the EU region. Okay, I'll keep scrolling down. Yeah, yeah. I think I scrolled up and there are a lot of questions here, as you said. Please share some ideal lab layout. I would request representatives of Kiwani to do the needful. The next question is coming from Mr. Puneet. Dear Madam, in case of any archival, there's a high possibility that the product may not come under the purview of regulators in the long run. In such cases, how long do we need to archive documents? Again, it varies case to case basis. In case you are, again based on the regulatory requirements of the submissions that you're doing. Like archival of data, are there any specific guidelines for data retrieval? This is by Mr. Alar Kulkarni. Here I would like to indicate that the entire process of data archival and data retrieval is designed by the relevant organizations, and they have systems in place. Now remote access is being given, and you have specifically said during the pandemic situation. So in such situations, remote access is being provided for employees to access data. In fact, we have done regulatory inspections handling remotely as well. Last week, I did a remote inspection for a facility I'm right now in Bangalore. I did a remote inspection or a virtual inspection for a facility in Andhra Pradesh. So they were able to upload the documents and I was able to do that. So there's a question from, there's a question regarding, are there guidelines for lab furniture selections? Again, coming in from Elark Kulkarni. Again, there are guidelines pertaining to the lab layouts and the way the material has to move. And again, lab furniture selection is dependent on the activity being conducted. Like for example, when you're handling microbiological samples or whatever, appropriate furniture has to be selected. And I'm sure the Kiwani engineers would be able to guide you in selection of lab furniture. So, and there is a question that what is the expiry date of analyst certification, being queried by Mr. Lakshman Kaswan. The analyst certification again depends based on the analytical methods that they're handling or the analytical techniques that they are handling, and also the organization's organization's metrics on training curriculum. So it's based on that the analyst certification happens. Typically, it is annual certification. I have come across, and I have also been an analyst. I was an enzyme analyst, and during that time, I was being certified every year. Every year I was given a blind sample or I was given an unknown sample, and I had to undergo the certification process every year. Do we have time for more questions? Yeah, I think the last question. Please don't worry, we would ensure that all the questions are adequately addressed in our communication to you. I understand one question being asked by Mageswari. I understand we need to follow GLP for labs. Do we need any certification for the labs? Again, I would advise you that it depends on the type of activity, the laboratory activity that you are doing in your particular laboratory. If in case it is preclinical, then you need to go in for GLP certification. In case it is clinical samples, you need to go in for ISO 15189 certification that is done by the National Accreditation Board for Testing and Calibration Labs. That is as per the ISO 15189. That is the medical lab quality systems. Okay. And so, and if in case it is any other testing lab, like for example, water sample testing or any chemical analysis, or if it's a metallurgy lab, then you need to undergo ISO 17025 laboratory certification that is again handled by the NABL body. I think we have to conclude. Thank you very much for attending the webinar.
Hey everyone, thank you so much. Thank you for attending. We are sorry that we couldn't answer all the questions, but don't worry. Just follow us on our social media channels, LinkedIn, or just keep watching our website. We will be posting the responses to your questions. Thank you again for joining in, and have a wonderful evening. Bye-bye.